Yeah, absolutely. Please.
Hello, everybody. Our speaker today is Dr. Julie Meachen. She's an associate professor of anatomy at Des Moines University and the lead investigator at Natural Trap Cave in Wyoming. She earned her bachelor's and master's degrees in zoology at the University of Florida and completed her PhD in ecology and evolutionary biology in 2008 at UCLA before going on to do a postdoctoral fellowship at the National Evolutionary Synthesis Center. Dr. Meachen studies paleoecology and functional morphology with a focus on analyzing how environmental changes, particularly global warming and extinction events, affect mammal species, particularly carnivorans, at the end of the Pleistocene. So with that, I'm going to hand off the microphone. Please give a warm welcome to our speaker.
Thank you. All right, so I've had a really fun time here today. Thank you guys for all being amazing hosts. I'm going to move this before I trip over it because I am not known for being graceful. All right, thanks. I hope everybody enjoys my talk. This is a new talk made up of lots of old things, but we'll see how it goes. All right, so, ooh, that's really dark. So my research program is generally composed of two overarching themes. The first theme that I like to focus on is the pre or historical biology of the group Canidae, which are the dogs. I don't know why, but I really like them, and mostly in North America. And the second overarching theme of my research program is how can we use fossils to examine patterns of faunal and environmental change from the Pleistocene to the Holocene. So looking at that important extinction time in Earth's history. And then, of course, whenever I can combine these two things, that is a big bonus for me. So let's start off by talking about my two main study sites. These two sites are different in a lot of ways, but they also have a lot in common. The main thing that these two sites have in common, and the two sites are Natural Trap Cave, which is the one over here. And then this one is the Rancho La Brea tar pits. They're different in that one is in the middle of nowhere, and it's very open and arid. The other one is in the middle of a major city, and the habitat was probably a little bit more closed during the Pleistocene. But the two things that they have in common is that they both are at the end of the Pleistocene. So they're both late Pleistocene sites with large mammals. And they both have a long enough fossil record to reconstruct the environment and change through time. So they do have some really important things in common. And some of you may have seen this kind of table before, but it's basically climate fluctuations through the late Pleistocene from about 50,000 years ago until the present. Temperature is there on that y-axis. And this is the time period that I study right here in this red box. And as you can see, there's lots of climate fluctuations. So I'm really interested in this time period specifically because we have some really major ones like the Bowling-Alarod interstadial, the Younger Dryas, and of course the transition from the Pleistocene to the Holocene, where we get that major jump in climate, and then we kind of settle out in the Holocene, which is way less, you know, variability than in the Pleistocene. So what I'm going to be talking to you guys specifically today is about two different things. The first one is going to be about my work on North American canids, and I've got a couple of different things I want to talk about with that. And then the other thing I want to talk about is the Pleistocene extinction event and climate. And so there are two sites, my two sites that I work on that I'm going to talk about with that. So let's go ahead and jump into some dogs because they're my favorite. And let's talk about Pleistocene North American canids in general. Most of what we know about Pleistocene North American canids comes from previous morphological fossil data. So we find fossils, we do morphological analyses, and that's basically how we know what we know about canids. And in North America during the Pleistocene, there really were 4 major canid species. That was it. There's not that many. There are dire wolves, which you probably have all heard of, right? I mean, even if you have never seen a dire wolf specimen, which you probably have, there's a skull cast in the library. You've probably heard of them on Game of Thrones. And so everybody knows about them from that. As it turns out, spoiler, I'm going to tell you something that Game of Thrones got terribly wrong about dire wolves. There are also gray wolves in North America during the Pleistocene. They are not very abundant, but they are here. We also have coyotes, which are abundant in the Pleistocene. And then finally, we have the elusive red wolf, which it seems today there's a lot of news about the red wolf not being a real species. People think it might be a hybrid between wolves and coyotes. As it turns out, our historic red wolves were something totally different than what we have left in North America today. So I'm not going to touch too much on that, but they were here in North America. So what I am going to talk about to begin with is dire wolf DNA. And we have never gotten DNA from dire wolves before this study came out. Everything that we knew about dire wolves was based on morphological data. And because dire wolves and gray wolves look so similar in their morphology, we assumed that the two were sister species. So we really didn't know where they were going to fall out with DNA. We were kind of hoping that maybe they would be sister species, but I don't know. So it turns out that in 2015 or 2016 and 2017, 3 separate groups of researchers decided they were going to do dire wolf DNA at the same time. And 2 of those groups actually came to me as the morphologist, like, hey, we're going to do dire wolf DNA. Will you be our morphologist? And I kind of was like, oh, wait, do you know that there are 2 other groups doing this? And they were like, no. So I decided that I would try to bring them all together into one big team to make one big dire wolf paper rather than several small dire wolf papers, which actually worked out because we combined the teams. We actually sequenced 5 different genomes from dire wolves from all over the United States. So we had one from Ohio, one from Idaho, one from Natural Trap Cave, which is my site, and one from Tennessee. And with these dire wolves, we were able to actually sequence mitochondrial and low coverage nuclear DNA from the dire wolf. So where are dire wolves? Well, there are coyotes and gray wolves right there. There's the dire wolf. So not closely related to the gray wolf at all. This was kind of a shock to us because as paleontologists and morphologists, we really do rely on the morphology to tell us the true story. And as it turns out, the morphology was not telling us the story, the full story here. So there's actually 5.7 million years of evolution between the dire wolves and the clade that's responsible for coyotes and gray wolves. So what's going on? What does this mean? So it means that dire wolves actually split from the lineage that has the living coyotes and gray wolves at least 5.7 million years ago. So that's a long time. During this time, we see no evidence for gene flow between coyotes and between gray wolves and dire wolves. So there's basically no genetic admixture in there. So they're their own thing. So dire wolves evolved in isolation in North America, at least from gray wolves. Maybe they were breeding with some other things that were extinct, but certainly not with gray wolves. While we think that gray wolves and coyotes are actually relatively newcomers to North America. So they basically came to North America late in the game for dogs. Because of this difference, this huge genetic difference, we decided as a group on this paper to change the genus of dire wolves from Canis to an Anoscion. And Anoscion was a genus name that was originally given to dire wolves by Miriam back at the turn of the last century. So basically we went back to its old name, so it's its own genus now. And so what does this mean for canid phylogenetics based on morphology? They're very confusing. They don't follow our rules, right? Like if you count their cusps, you're not going to get the right answer. So I'm going to come back to this in a little bit about what this means and how we're going to sort out canids in North America at this point. But this was a reconstruction that Mauricio Anton did for our Nature paper in 2021 when it came out. And this is at Rancho La Brea, and it's a group of dire wolves eating a bison and chasing off a couple of stray mangy gray wolves. And so in the past, dire wolves have always been depicted as being like a gray wolf on steroids, right? Like it's a big fluffy wolf, right? So Game of Thrones, the dire wolves are the wolves of the north. No, actually, as it turns out, the dire wolves are the wolves of the south. Dire wolves actually are super duper rare above 55 degrees north latitude. And so we kind of tried to depict them with this sort of reddish short hair, kind of a little bit reminiscent of like a dhole, right? An Indian dhole, because they really are a warmer adapted species. So they're not going to look like the big fuzzy gray wolves that we have running around now. So we just wanted to give a different view of a dire wolf based on its ecology and its genetics. So this leads me to my question that I posed before, New World canids. We thought we had a good idea of who is related to who, but not so much now. Many canids of unknown relatedness. So we have Canis armbruesteri, which is Armbruster's dog, which we thought was possibly related to dire wolves. And we also have this thing called Canis lepophagus, which is a small dog that existed like in the Pliocene. So about 3 million years ago, we thought this was a coyote predecessor, but if coyotes and gray wolves are relatively newcomers to the New World, is this actually related to coyotes or is it more closely related to dire wolves? We don't know. So I'll come back to this later when I'm talking about future projects that I want to do on dogs. So let's kind of switch from dire wolves to the wolves of Natural Trap Cave. So when I went to Natural Trap Cave for the first time in 2014, I found this canid that was wolf-like. It looked like a wolf, but it was kind of small. It was smaller than I would expect for a dire wolf. And the original excavators at Natural Trap Cave basically said, oh, well, we have coyotes and we have dire wolves. And I was like, okay, well, we have coyotes and we have dire wolves. So I was like, is this a coyote or is this a dire wolf? And I didn't know what was going on. And so I decided to throw some geometric morphometrics at it. I'm like, I'm going to figure out what's going on here. So in 2016, I did this study where I basically looked at the morphology of the jaws of the wolves from Natural Trap Cave. And I compared them to other canids that were around. So these blue circles up here are all dire wolves. These green squares up here are all modern gray wolves. So these are from modern populations. And then these red triangles down here are these things called Beringian wolves, which it turns out are actually, they're related to gray wolves. So they're a gray wolf morph, but they existed up in Alaska and the Yukon and in Siberia. So they're basically a New World, Old World species, and they only we only find them in the Pleistocene, so they're not around today. They're basically like the Arctic version of the dire wolf, although they're closely related to the gray wolf. So this would kind of be more like what really should be in Game of Thrones would be the Beringian wolves. So anyway, I said, well, where are the Natural Trap Cave wolves? So here they are. There are these purple diamonds up here. And when I did some statistical analyses on these morphospaces, what I found is that the Natural Trap Cave wolves actually group with the Beringian wolves. So they group with these gray wolves rather than dire wolves. Well, we know we actually do have dire wolves at Natural Trap. We got genetic data from one, but it just turns out they're in very low abundance. So they're there, but they're rare. So I thought, okay, well, these are Beringian wolves, right? Let's see if we can get some DNA from them. So the canid conundrum at Natural Trap, who is there? Dire wolves are present, but in low numbers. And we've managed to sequence 8 other canids from Natural Trap Cave. And of course, I didn't do this myself. I worked with a team of people who do dog genetics. And what we found is that the mitochondrial DNA of these critters, which we get quite a bit of, looks a lot like a coyote. So this is a clade of coyotes. We actually have mid-continent coyotes here, so things that are like, you know, here in Minnesota. We have eastern wolves and red wolves here. This is what their mitochondrial DNA looks like. This is California coyotes. And then down here, sort of far removed from these other things, are the coyotes, or the coyotes are the wolves from Natural Trap Cave. So the canids from Natural Trap Cave, I should say. So we were like, oh, well, we have coyotes from Natural Trap Cave, right? Like these are coyotes, they're really big coyotes. Well, we finally did get nuclear DNA, and our nuclear DNA says something totally different. So here is where our Natural Trap Cave wolf falls. We only have nuclear data from one, but if you look on this graph, these are coyotes over here. These are all wolves. So our Natural Trap Cave specimen doesn't have coyote nuclear DNA. It's not a coyote. So what is it? So although the mitochondrial DNA is closer to coyotes than to modern gray wolf, it's pretty clear from our phylogeny that it's actually distinct from the living coyotes. So it's not a living coyote, it's not a modern coyote. And the nuclear DNA shows they're wolves, but not closely related to modern populations. So what we think is happening is that there's some admixture going on. They share some of their nuclear DNA affinities with those Beringian wolves from Alaska and the Yukon. And this is basically where Natural Trap Cave is located, right here. So this is the Cordilleran ice sheet and the Laurentide ice sheet. When these two thawed enough to form this corridor, it led directly from Beringia to Natural Trap Cave. So basically Natural Trap Cave is like the chute through from Beringia. And this is not a huge shocker because we've actually found at Natural Trap Cave, we've sequenced bison DNA, we've sequenced muskox DNA, and both of them have affinities from Beringia. So they both basically came down from Beringia. So what we think is happening at Natural Trap Cave is that we're getting Beringian wolves coming down to Natural Trap. We think we have some native North American canids in the lower 48 already. Remember, canids arose and evolved in North America. They're a native North American group. So we've already got canids here. And what we think is happening is we have some native North American canids. My collaborator, Ben Sachs, thinks that they are red wolves. So he thinks the red wolves that were native to North America admixed with the gray wolves coming down from Beringia, and that's what we have at Natural Trap Cave. So he thinks that's what we're getting. I wish we could get better nuclear DNA from these guys, but it's really hard. The preservation is not awesome all the time. And so I'm kind of just holding my breath, hoping that we find something good. But we are hoping to publish this paper before I go back into the field. So it's going to be what it's going to be. But it's a really cool project too, because this is the first ancient DNA from a wolf in the lower 48 states. Like we've never gotten ancient DNA from a wolf in the lower 48. We've gotten tons from Beringia and Alaska, but nothing down here. So it's our first example of what 44,000-year-old gray wolf DNA looks like in North America. So here's my question that came from all of this. If dire wolves are not related to gray wolves and coyotes, when did coyotes arise from gray wolves? Like when did they separate from gray wolves or speciate? They maybe didn't arise, but the two species split. So they're sister species, coyotes and wolves. And gray wolves do exist in low densities in the continental US before the end Pleistocene, but we know that they came over from Eurasia. So gray wolves did, for sure. Gray wolves are common in Canada and Alaska in the Pleistocene, but not in the lower 48. When did coyotes arise and where? There aren't coyote fossils in Beringia or Eurasia. Like there's like one coyote ever found in Beringia and there's none in Eurasia. So when did they arise? And And more importantly, where are the fossils outside of mid-continent and southern North America? There's none in the northern region. And so one of the things that we're actually exploring is that coyotes may be descended from red wolves instead of gray wolves. So this is one of the hypotheses that we're kind of playing with, because as we know, red wolves were different in the Pleistocene. So before they basically mostly went extinct and then admixed with coyotes again, it's possible that they were a separate group that was already here. So the answer is we need more ancient DNA from coyotes and any Pleistocene wolves. So this has kind of been my goal, is to like find all this coyote DNA. And I've been kind of scouring museums looking for coyotes that people will let me sample. Can I sample your coyotes? Can I sample your coyotes? So this has kind of been my crusade for a little while. We'll see how it goes. So canid relatedness, a doggone dilemma. Who is related to who? So as a consequence of the now known ancestry of the dire wolf, there are many fossil species who now have a dubious place in the canid tree, which we mentioned before. How do we figure out where they belong? I don't think ancient DNA is going to cut it. These things are older than Pleistocene. They're Pliocene. They probably don't have DNA in them anymore. So can we look at them with fresh eyes and do a morphological analysis, knowing that dire wolves and gray wolves are not related? Maybe we need to do morphological analyses of those two species again, keeping in mind that they are totally different. I don't know if that's possible, but it's something that I would like to kind of pursue a little bit. So with that, I'm going to switch to actually probably my favorite canid. Dire wolves are cool. They're so sexy. Gray wolves, you know, they're awesome. Some people hate them, but I like them. But coyotes have a special place in my heart, and you're about to find out why. They are the most widespread large carnivore today in North America. And I say large in quotes because they're really not large. They're somewhere between, 20 to 25 kilograms, maybe a little less, maybe like, 15 to 25 kilograms, depending upon where you are. But they are the most widespread megafauna in North America today. They have a labile community structure, meaning that they are able to change their ecology and their family groups depending upon their situation. So, for example, when there are a lot of wolves around, they actually will increase their pack size, mainly as defense. And as a consequence of increasing their pack size, they can do things like hunt deer that they would not do if they were smaller or if they were more solitary, right? If they're alone, they can hunt rabbits, they can hunt rodents. But if there's like 10 of them, they can take down a deer. So they do change their ecology based on their situation. And following to that closely is that they're dietary opportunists. They can eat both large and small prey. They can actually eat, they can be omnivorous, they can eat other things. They can pretty much eat anything, including garbage. Coyotes, yeah. And see, they can coexist quite well with humans and have established communities in large urban centers. So for example, there's a huge population of urban coyotes in Chicago, and there's a huge population of urban coyotes in Los Angeles. I have friends that live in both cities, and they're like, walked my dog at 10 and saw a pack of coyotes in the road. I was like, yeah, you probably don't want to mess with them. All right, so how have coyotes evolved over the last 40,000 years? What's happened to coyotes? I got a wild hair, I want to know. So this was a study that I actually did when I was a postdoc at Nescent. I had measured all these coyotes for something, and I looked at them and I'm like, these are kind of weird. So I was like, what's going on? So I decided to look at these coyotes from Rancho, La Brea. This is a subspecies of coyote that only existed in the Pleistocene. It's called Canis, latrans, or cati. And these ones are from La Brea. And so this is a modern coyote skull, and this is a coyote skull from Rancho, La Brea. And so what I did was I actually measured all the postcrania of these coyotes. I had several different populations. I measured some from Rancho La Brea, I measured some from some other Pleistocene sites in California. I measured them from a pit at Rancho La Brea called Pit 10 that is about 10,000 years old. So it's basically deposited right after the extinction event. And then I measured coyotes from a couple of Holocene caves that are about 4,000 years old. And then I measured modern population. So I pretty much got coyotes through time. And I wanted to see how they changed. So I'll give you the results in a second. This is the second study I did on the jaws of coyotes. And I wanted to look and see how their jaws have changed over time. And so I placed landmarks, 2D landmarks on these coyote jaws to give me an idea of a couple things. This is a great indication of premolar length. So basically the shearing teeth. We have the molar arcade here, which is for grinding. And then we have a few other things. We have jaw depth with these points together that can give us a good idea of, what are the mechanical stressors happening to these coyote jaws. So these are the results from the postcranial study that I did. And this was a study I published in 2012. This is femur circumference here on the y-axis, and femur circumference is a really good indicator of size. So how big are these individuals? And what we find here is that in the Pleistocene, coyotes are big. They're really big. So I would estimate their size at somewhere around 25 kilos in the Pleistocene. That's probably average coyote size. After the extinction event in Pit 10, so this is at the same site, but just after the extinction event, coyote body size decreases significantly. So this is a significant value. And that continues throughout the Holocene. So basically they stay small. So we see this huge body size change. What's going on with this? We'll look at the jaws and then we'll figure out the whole picture. So here are the coyote jaws. And the results of the coyote jaws, the ones in the upper left-hand corner there, those are the Pleistocene coyotes. So Pleistocene coyotes are in blue. These green circles are modern coyotes. And these black triangles are coyotes from Pit 10. So Pit 10 is that pit that was deposited directly after the extinction event. And they actually all occupy different places in morphospace. And you can't really tell very well, but I tried to highlight in these little images the differences. Up here, this jaw has pretty big teeth and it's actually pretty robust. And I'll illustrate this in my next slide. This is the modern coyote. The jaw is much thinner. The teeth are more gracile. There's less shearing. The shearing arcade is smaller and the grinding arcade is larger. And then in Pit 10, what it looks like we have the Pleistocene complement of teeth in the modern style jaw. So basically we have big robust teeth in a tiny skinny jaw in Pit 10, which I thought was really interesting. I'll speculate on why that could possibly be. I think that the bony structures probably change faster than the teeth. And so we might be getting sort of a mosaic evolution happening, but I don't know that for sure. That is just simply my guess. So here's what the differences in the jaw look like. So when I measured the shearing arcade relative to the grinding arcade, what we see is that in the modern coyotes, we have a smaller shearing arcade, a larger grinding arcade. And in the Pleistocene coyotes, we have a relatively smaller grinding arcade to shearing arcade. So for those of you who are unaware what those two different things mean, the shearing teeth are going to be for flesh ripping. So they're basically going to be an indication of how much large carnivore meat they are eating. So they're going to be shearing with those teeth. The grinding teeth can pull double duty. They can be used to crush bones. So it might be an indication of how much scavenging is going on, but they can also be used to grind up fruit and things like that. So it might be an indication of omnivory. So either or. And then what we see here is this arcade at the front of the jaw, the anterior jaw depth is changing. It's getting smaller in the Holocene coyotes. This was kind of harder for me to figure out, but I finally read this paper that was by actually my dissertation advisor and one of the students on her committee, and this was a while ago. And what they found in large canids who hunt large prey, especially African hunting dogs and wolves, is they both have this behavioral adaptation where when they run after prey, they chase it and they wear it down, but they do this behavior where they actually ram their face into the flank of the prey to like wear it down. So they actually like smack them with their face and they run the front part of their face in there. And as it turns out, all of the big dogs like wolves and hunting dogs that hunt these big prey have a deeper front jaw because of the forces of that behavior, smacking their face into the prey. And also because when they're chewing on like hides of big ungulates, right? Like they've got this thick hide, it basically strengthens that symphysis so that buttresses that area. So what it looks like to me, what's going on with these coyotes is that they are changing in size, but they also might be changing the prey that they are killing between the Pleistocene and the Holocene. So in the Pleistocene, coyotes coexist with many different competitors, lots of other carnivores, right? So they might be getting competitively excluded from certain things in the Pleistocene, but they also coexist with many more prey options. And I am not suggesting that coyotes were killing bison or horses, but those things do have babies, right? And they are definitely much more bite-sized. So it is possible that coyotes probably took advantage of some of that during the Pleistocene. We know they hunt in groups now, right? When there's other big predators around. So think about that. They probably hunted in bigger groups in the Pleistocene. And we do have evidence for that too, which I'll tell you in just a few slides. So it looks like they probably switched from taking larger prey to smaller prey at the end of the Pleistocene, which makes sense. So both of these things could affect their morphology and ecology. And as we know, coyotes are labile. There's no reason they didn't change after the Pleistocene. So how did megafaunal extinction affect this modern species? Probably coyote, well, we know that coyotes became smaller. Probably they switched from larger prey to smaller prey and became more solitary in the Holocene. So the conclusions I got from this study is that its extinction events don't occur in a vacuum. Even though coyotes lived through the extinction event, they didn't come out on the other side without changes. And you know, now coyotes are living in cities, right? So they're city coyotes, and they also go to Quiznos and ride the subway. So you know, coyotes are going to take over the world. It's just the way it's going to be. They live with us just fine, so why not? All right. Anyway, so I'm not done with coyotes yet. I'm still looking at coyotes. And so after all that work that I did on coyotes on my own, I'm now part of a big research team at Rancho, Brea. And we're doing this huge grant where we're doing like a massive dating of the Rancho Brea tar pits, which is really cool because not only is it giving us some really cool ecological data, but it's also increasing the number of Pleistocene dates in North America by like threefold or something. Like we're basically tripling the number of radiocarbon dates in North America at the end of the Pleistocene. So what are we doing with this project? We're examining the tar pits as an ecological site using basically a whole bunch of different techniques to answer questions about the end Pleistocene extinction event, how climate changes megafauna or affects the megafauna, and to examine ecological changes in both extinct species before the extinction event and extant species through the whole event. And coyotes are our extant species. And the reason why we chose coyotes is kind of based on my previous work, but also because they are the third most common species at the tar pit. And this kind of is the data that lends credence to the fact that coyotes are probably hunting in big groups in the Pleistocene. The reason why is because we know that the tar pits are a carnivore trap. So the species that's most common is dire wolves, followed closely by the saber-toothed cat, Smilodon fatalis, and then followed third by the coyote. Well, why are these two species in particular so abundant at the tar pits? That was the subject of a study that my dissertation advisor and a lot of others did. several years ago, and they wanted to find out why those species in particular were so numerous at the tar pits. So they actually did this really awesome study where they set up these playback experiments in the African savanna. They played noises of carnivores fighting and of ungulates dying. So basically like screaming and death noises of ungulates and carnivores fighting. And they put up camera traps to watch who came. And guess who came? The big group living carnivores. So they were all like, yeah, I'm getting my boys, I'm ready for a fight. Let's do it. So basically lions, hyenas, hunting dogs. So the only ones that came to those noises. Leopards, cheetahs, other solitary predators ran away. They're like, I'm getting out of here. So what we think happened at the tar pits is that an herbivore would get stuck. And it would basically be like the African savanna playback experiment. It would be smorgasbord, right? Like the herbivore would be like, get me out of here. And the screams of the herbivore would attract the big group carnivores. So Smilodon, direwolves, coyotes. These are the ones that have their little posse, and they're going to come and they are going to get rid of anything else that's there, right? Like they're going to hold their own because they got their boys and girls with them. So that's why we think coyotes probably were social, and they had big groups in the Pleistocene, is because if they're going to come where there's direwolves and Smilodon, you better bring a bunch of you, right? So there you go. So coyotes are found in the Pleistocene and the early Holocene, and they're still around in Southern California. So we have a great record of them. So in addition to the morphological data that I collected back in 2011 for that paper, we collected some new data for coyotes. We collected data on 136 coyote specimens from 5 different deposits at Rancho, La Brea, and 16 modern coyotes from the Santa Barbara Museum. So definitely a similar type of environment, fairly close by. And all the coyotes that were from the Santa Barbara Museum were less than 100 years old. So 2 things in particular that we collected that were really cool are we collected radiocarbon dates. So we have tons of radiocarbon dates spanning 45,000 years to about 765 years before present from La Brea. And then we have those modern dates. And we collected stable isotopes from the mandibular bone collagen when we did the radiocarbon date, which give us delta 15 nitrogen and delta C13 carbon. So nitrogen is really cool because generally when you think about nitrogen values and stable isotopes on animals, what you're going to be looking for is trophic level. So you're going to basically be looking for how much protein does this animal ingest. Well, here we found. So generally, this is nitrogen over here on the y-axis. This is time on the x-axis. Generally, the higher the nitrogen values, the higher the trophic level. So basically, when your nitrogen values are high, it means you eat more meat. There are some exceptions to this, of course. Marine mammals are an exception. But for coyotes, it generally holds true. So this red line here is the extinction event. And it kind of looks like 2 different data sets, doesn't it? It's like, oh, coyotes are kind of hanging out, and then like, boop, they fall off the cliff. Their nitrogen values go into the dumps. And we're like, whoa, this is big. Like, this is a huge result. And it's a significant result. So the Pleistocene delta nitrogen value is 11 parts per mil. Holocene average is 7.8 parts per mil, and modern average is 8. So this is a significant result. It's a big deal. So what's going on? Are coyotes going through a major trophic shift? This was our initial thought. Oh my God, they're changing their diet. They're starting to eat more plants after the Pleistocene, right? Well, as it turns out, we took data on pumas through the extinction event too, and they show the same drop. And we're like, ain't no way puma's eating fruit. So what is going on? So we took all of these samples that are circled here in yellow, And we sent them to a lab that actually breaks down nitrogen into all of its amino acids. And this is what we found when we got a value for the trophic position. So basically, trophic position is calculated by two different amino acids, gluthionine and phenylalanine. And what we found is that trophic position has not changed in coyotes. They ate meat in the Pleistocene, they're still eating meat now in the Holocene. So it's not trophic level. What we found is a huge decline in phenylalanine. And phenylalanine is an essential, a source, an essential amino acid, meaning they have to get it from their diet. So it captures the entire food chain. So herbivores have to get it from their diet, carnivores have to get it from their diet. So basically what this means is a major baseline shift in the nitrogen values at the end of the Pleistocene. So something is happening in the environment. So it is not a trophic level shift, but it is basically a decline in nitrogen due to a baseline shift. So this means plants are depleted in nitrogen at the end of the Pleistocene, and so are animals. So this is pretty cool. Our previous hypothesis was that coyotes declined in size from the past to present, and with a bulk nitrogen value suggestive of a more omnivorous diet. So that's what we thought. Our revised hypothesis is that there's a baseline shift. Why is there a baseline shift? It could be an increase in fire frequency and intensity. Could be a decline in megafauna that leads to different changes in nitrogen cycling. Could be changes in precipitation. All of these things are possible and lead to nitrogen baseline shifts. So let's take it a step further and see maybe what is going on. So what do coyotes tell us? They're cool. They're the bellwethers of the Pleistocene-Holocene transition. The transition from the Pleistocene to the Holocene was actually recorded in their tissues. Like they showed us what's going on. A follow-up question I have is, was this major state shift occurring everywhere? Like, do we have a nitrogen baseline shift all over North America? We need more coyote data, and we need more data, period. So what's causing this baseline shift? Well, it turns out the next study I'm going to tell you, which is about Pleistocene extinctions and climate, might actually be able to answer the question. So let's go back to the Rancho La Brea tar pits. We're going to look at some extinction. Rancho La Brea is thousands of fossils that date somewhere, between 50,000 years old and the present. It spans major climatic events, including the last glacial maximum and like the bowling alley rod, all of these major changes in Earth's history, the extinction event. So we did the 400 new dates. 172 dates of those span the extinction event. So they basically come right up to the extinction. And here is the data that we found. So when we did all these dates, we have all these Pleistocene species. We have sloths, we have camels, bison, horses, American lions, dire wolves, Smilodon, and here's coyotes. So basically, these red lines indicate the last known date of each of these species at Rancho La Brea. And coyotes, of course, continue through. So these are all coyote dates past the extinction event. But basically, all extinct taxa together end up going extinct right here around 13,000 years before present. So that's basically the extinction event captured at Rancho La Brea. So what's going on here? Well, with only our data, we noticed kind of an interesting pattern. One of this pattern is all individuals together, all species together. We see this gradual decline here, but when we actually break the data down by taxa, we find that there are two pulses of carnivore decline and one major pulse of herbivore decline. So our sample size might actually not be big enough to be all that statistically significant, but it's a really interesting finding. And we see that herbivores are declining, and then carnivores are declining. So basically, we see this trophic cascade. And this first pulse actually includes things like camels and some bison. And the second pulse is mostly horses and bison. So camels actually tap out before everybody else at Rancho La Brea. So when we added a couple other data points, I know this is kind of small and you can't really see it, but two major things that we added are we added pollen to this. And we added charcoal data, and I really want to highlight the charcoal data. Here it is blown up. This red line here is the extinction event. This is a charcoal spike. So we have this huge charcoal spike right around that time when all these animals went extinct at La Brea. So what we really thought was going on is we had a concentrated charcoal spike that coincided with the extinction event. We know this. Humans were known to be in mid-continental North America by this time, although admittedly the fossil record of humans in Southern California is very poor at this time. However, due to some other data that our team had collected showing that humans caused these kind of fire spikes, I believe in Australia and a few other localities where we know they were causing them, our most parsimonious conclusion is that humans are likely responsible for these fire spikes because they're pretty sustained. Of course, there are other possible causes, lightning, etc. So it's possible that they were caused by other things, but humans are the most parsimonious answer. Okay, and now my thing is, oh, did it? Nope. Oh, let me go figure out what's going on. I'm having technical difficulties. Apologize. Okay. So we have this concentrated charcoal spike before and during the Pleistocene extinction event. That coincides with the megafaunal. Oh, I already did that. Okay. Extinction event, likely a 1-2 punch of climate change and habitat restructuring. So basically what happens is these fires spread. They basically destroy a lot of the habitat that was there. And so when the flora is destroyed, the fauna can't eat, and this leads to a cascade where things go extinct. So basically, we think that these things came together in this 1-2 punch. So not overkill, but humans, right? Humans modifying their habitat, which we're really still good at doing today, right? So we have an idea of what happened in Southern California. We're collecting more data to determine what happened at Natural Trap Cave, although this might be difficult because we're actually missing the extinction event at Natural Trap Cave, so that may not end up working. Hopefully we can figure something out. Both of these sites are in the Western US, but what happened elsewhere in North America? How did the extinction event happen? Did it happen the same everywhere? Did it happen different everywhere? What other sites, especially from the Eastern US, have the type of faunal record that can help answer this question? And that's one of the things that I want to look into. So some future work that I'm doing at Natural Trap Cave, kind of going along with this extinction event, is actually some work on aridity, so drying. Our overall goal for this project that I'm now embarking on is to use the late Pleistocene fossil site of Natural Trap Cave to examine the effects of aridity basically over the last 39,000 years. So we're going to use modern fauna to better understand how different species respond to aridity. And we're going to use different fauna who have different sensitivities to water. So basically species that need to drink water and species that don't need to drink water. We're going to use stable isotope analysis of sediment and organic matter to get a good climate proxy for the site, as well as products from bacteria. And we're going to use these things to determine how the fauna at Natural Trap Cave responded to these fluctuations using both morphometrics and isotopes. So basically looking and seeing, is their morphology changing? Are their chemical values changing? And we have some preliminary data that we're putting in a grant. And so some of these things include, here's our precipitation record at Natural Trap Cave. We've got a pretty good record in the late Pleistocene, early Holocene. Here is our pollen data. We actually have much better resolution on pollen now to recreate a climate record. These are bacterial branched glycerol diacyl glycerol tetraethers, which I don't work on. So I'm sorry if I got that wrong. So we actually have a postdoc. This is what she does. She works on these things. Evidently, these are byproducts from bacteria that actually can record climate in the soil. So that is really cool and interesting. And we definitely have a better record of this now. Our new data is not smooth, thank goodness. And also we have hydrogen values from sediment, and these are oxygen values, or sorry, carbon values, that both record some of the temperatures and precipitation at natural trap over especially the whole Holocene, the entire Holocene. So our hope is to submit this grant and get this grant and then do some really cool climate science at Natural Trap Cave. So with that, I just want to thank all my hosts here, everybody who's met with me. I had a really great time today. And I want to thank all my collaborators from Natural Trap, from Rancho, Brea, and from all my canid work. So just thanks for having me. I'll leave you with fun field work pictures. And I'm happy to take questions if I have time. Yeah. Mhm. Yeah, and that's cool. We did do a PCA of that. That was that's my collaborator Kieran Mitchell who did that. And that is kind of I think our whole group, our ancient DNA wolf group, are they've done some of the geneticists have used that technique before, and I'd never seen that done either. So I thought that was really super cool. Yeah.
Great talk. I was just wondering if with especially I wonder at La Brea they have this, but these larger packs of coyotes that you've seen.
Yeah.
Is there any indication or has anyone done anything on like any kind of caste system dimorphism? Like, is there any kind of like, because I know with Canis and some other groups where there's like these more complex hierarchies going on, that you see dimorphism as well. Is anyone?
So that's a great question. As it turns out, canids have a pretty simple social system. The whole alpha thing is not really real, right? And so canids are mostly like family groups. So they're relatives generally. And so when in good times, it's usually just one adult female and one adult male, and then their year-old pups, basically helping raise the next litter of babies. But in hard times when they have to like deal with other wolves and stuff, it can be like sisters and brothers banding together, and youngins staying in the pack later. So generally there's not. And then even with canid, sexual dimorphism is actually reduced compared to other carnivores. It's there, absolutely, but it's not like big cats or bears.
And I guess the other question I had was when you're talking about the baseline shift in those nitrogen,
Yeah.
right? And you saw it across coyotes and pumas. I was just curious with the coyotes, if you think like occurrences like that where these major environmental shifts, do you think that would aid in their like labialness, I guess, further?
Possibly, right? I mean, if you're selecting for an individual or groups of individuals who are able to make changes, it's possible that it could have, right? If they managed to make through the extinction event and still eat and survive, it could select for the individuals that were able to make those changes quickly.
Yeah, I'm just wondering if like there was a specific, like not event, but shift like that started that kind of adaptability.
So one of the things I didn't talk about actually is we do see a size shift in coyotes, which I showed in my other work, but we actually, with the radiocarbon dates, actually were able to pinpoint exactly when that shift happened. It happened around 20,000 years ago. So something happened 20,000 years ago and coyotes started getting smaller. Like we can actually see their size go down with the radiocarbon dates. So I don't know exactly what happened then. My hypothesis actually was that gray wolves moved in to Rancho La Brea and things happened, like, you know, they got smaller because they had different competition. But we don't know. That's yeah, something happened at 20,000 years ago when they started getting smaller. Yeah.
How does your work on dogs compare to corresponding work in other major taxonomic groups?
Like mammals or just anything?
Cats, elephants.
So those are all mammals. So this work on dogs, specifically on morphology, ancient DNA. What did you have in mind?
Or is work in other taxonomic groups way ahead, way behind?
Okay, gotcha. So with morphology, dogs are pretty well studied. I mean, they've kind of been one of the groups that have been beaten to death with morphology. Doesn't always get right. With DNA, canids are ahead of some groups and behind others. So, with obviously with study species like common study species, we don't know much about canid genetics compared to like Drosophila or something like that. But in terms of modern carnivores, canids probably we probably know more about canids than we know about many other carnivores. For ancient DNA, I would say that canids and felids are both sort of like they're sexy and they're interesting and people want to study them. And so, people run that ancient DNA before they'll run something for like bighorn sheep, right? So they're kind of leading that field in terms of like Pleistocene DNA. I would say the one group that has more sequenced DNA for ancient DNA is Proboscideans. Like those have been sequenced way more. We found, and that's because they found a lot more of them in Siberia. They found these like well-preserved carcasses. So that's where I would put it generally. Yeah, Pete.
Yeah, so one thing that's kind of curious is, so you have these dogs coming in from Brea, and they're piling up in Southern California, at least that's what we're finding.
Yeah.
Then you have an entirely separate monophyletic group of South American canids. They're quite distinct.
Yeah.
Why do none of these wolf or wolf-like things make it to South America?
So that's a great question. So the answer is dire wolves do. We have dire wolves in South America. Coyotes do. So coyotes are in South America. They're not in southern South America, but they're in, they're in Mexico, and then they make it down a little bit further, and that's about where they stop. That's a great question. There are some Pliocene and early Pleistocene canids in South America that we think are also in North America. So some of these older things do make it to both continents. But gray wolves make it to Mexico, and that's it, right? We have Mexican wolves, but we don't have gray wolves in the rest of South America. So I don't know what the evolutionary, and the problem is that the fossil record in the tropics of South America is terrible. We have a good fossil record outside of the tropics for canids, but not where a lot of these canids live, like Spiothos and stuff like that. So they've figured things out with genetics as best they can, but they just don't have the really awesome fossil record. This microphone's giving me a lot of feedback. Sorry. Oh, that's why. Thank you. I'll move over here. They just don't have the really awesome fossil record like we have in North America. And I think that's probably in areas that are not tropical. I think it's probably just due to the fact that they just haven't been as well explored, right? So we might have good fox fossils, for example, because there's all those foxes, right, that are in South America that actually live in areas where there probably are fossils. So there are some, but I don't, yeah. Catherine.
You hinted at wanting to know what was going on in other parts of North America, like your two sites.
Yeah.
Are there known sites that you'd like to start exploring, or does there need to be totally new fieldwork to find new sites that would have the kind of record that you need?
That's a great question. There are some places that have known places. There's a bunch of places in Texas. So Texas has a great fossil record. Florida has a great fossil record for this time period. In the Midwest, we do have a fossil record for this time period. It's not as good as in those two places, but the Midwest would be another place to look. I was having this conversation with somebody earlier, and basically, as it turns out, most of the state of Iowa is private land. Like there's very little public land, and that's part of the problem, right? Like these fossils might be being found and then they're just being kept in people's houses or being sold. So they're not available for study. And so it would be great if we could find more Midwest sites that have like big assemblages. But right now, most of the really good sites are like Florida, Texas. There are some good ones in Tennessee. So those are kind of the places to go next for the Pleistocene stuff. Yeah.
With regards to those charcoal deposits in Brea, you mentioned immediately after that you were interested in aridity and natural trap.
Yeah.
Have you done, or has anyone done any work on aridity at the Brea around that time to see whether that might include or seemingly large scale fire events rather than these, since fire is pretty normal, but it's clearly there's something larger going on.
Yeah. So that charcoal actually wasn't from La Brea. It was from Southern California, but not La Brea. That's a great question. And the folks that work there, Reagan Dunn and Emily Lindsay, are actually looking at doing something like that as best they can. Reagan Dunn just started at the tar pits a couple of years ago. I think she started during the pandemic. And so she's really just starting her research there. And she's hopefully going to try to look at all sorts of different sediments. I think that the part of the problem with La Brea is that a lot of the stuff collected at the turn of the last century, like the 19 teens, they just pulled out the big sexy stuff and threw everything else away. Like they just didn't collect the stuff that we know is important today, right? And so some of those things have a good, like there are some deposits, pit deposits that have good data and some that don't. The ones that are being excavated now, all that data is being kept. So there are people looking at the microfauna and those kinds of records from this big deposit called Project 23. But Project 23 doesn't happen at the end of the Pleistocene. It's like, 40,000 years old. So I think they're pursuing those questions deeper at La Brea, but they may or may not have the fossil record to do it there because of the way things were preserved back in the 19 teens. Yeah.
When you're seeing the charcoal deposits
Mhm.
and you're seeing the loss of so many megafauna, the charcoal is seen at the exact same time or preceding or just after?
Just before and during.
But
Yes.
The camels before.
The camels went before, and the camels clearly were, the camels were kind of like on their way out before everybody else. And I don't know why. The habitat's already changing clearly. And I don't mean to suggest that the fires were the only things changing the habitat. We already were experiencing climate change. So things are changing and we get more dry species at the end. So horses, which are going to eat like hay and all the stuff that, nobody else wants to eat. We see those at the end. We see a lot of horses at the end and we see none of them when the camels are there. So basically there's clearly two different niches going on, two different habitats at that time.
Some megafauna that act as some species keeping down smaller brush type flora, and then when you lose them, then it grows up enough that fire spreads, whether it's lightning or humid fire. And
Yeah, yes, you're correct. So I would suggest though that it wouldn't be camels that would be doing it. I would suggest it's going to be bison and elephants that are going to be those two keystone species because they do modify their habitats no matter where they are. But those two, we don't have a great radiocarbon record for proboscideans at La Brea, which is weird because that's usually what the record is everywhere else. But we don't have a great record for those. But bison actually are one of the last things to fall off. So you're correct, and it could be that we don't know. But just with the data that we have, we don't think that is probably the explanation for what's happening at La Brea. Yeah.
Do you have a significant data set of herbivore Delta 15N, like thinking about that baseline shift?
Yeah, so we do. Did I just turn this? No, okay. We do. Herbivores are not as plentiful as carnivores, right? So we don't have the numbers of herbivores that we have of carnivores at that end Pleistocene extinction event, but we have nitrogen from all of those species that we looked at. So basically everything that you saw up there, we have nitrogen for.
You are seeing a baseline shift in as well.
We have not looked at that data yet. So the answer to your question is, I don't know. That's basically our last paper, is like this giant capstone paper of the project where we're basically going to be looking at all of this ecological data for the entire deposit and basically like dropping this giant packet of data. But the person doing our isotopes has not shared that data yet. So I do not know. Yeah.
So talk about this shift in body size that's likely occurring currently with the loss of a lot of their like available megafauna.
Mm-hmm.
Do you see a similar distribution in like contemporary coyote sizes, like in areas where there's, you know, whitetail versus mule deer, like, you know, size distribution?
That's a great question. And actually what we really see is we see bigger coyotes up in the northeast, but as it turns out, they're hybridizing with wolves up there. So I don't think it's actually due to some food source. I think it's just due to them having big genetics now. So big genes. Yeah, that's exactly how it happens. The genes are bigger and so the animals are bigger. No. So anyway, yeah, so they have big dog genes now. They have big dog genes. But I've looked at coyote body size all over North America, and they don't follow Bergman's rule. Let's just put it that way. So anybody who doesn't know what Bergman's rule is, it's that body size increases as you increase in latitude. So as you get further away from the equator, body size gets bigger. Coyotes don't do that. So their body size actually is really variable depending upon where they are. Like coyotes in California are slightly smaller than coyotes elsewhere, but there's not a temperature decline in coyote body size. So the only real body size change that we see in coyotes is when they hybridize with wolves, which of course is not due to ecology, but the fact that they now have wolf genetics. Yeah. Yeah. The diet could, yeah, of course they could take advantage of like fish. But for you mean for the nitrogen shift? So we thought about that, and the amino acid, the individual amino acids basically say, no, that's not what's going on. Yeah, that it's the plants. Yeah. Any last questions? I think we're probably over time at this point. So thank you, everybody. I really appreciate it.